Pasteurization is a heat treatment process that destroys pathogens in foods and beverage, notably milk, wine, and beer. This process is named after Louis Pasteur, a French scientist who is considered the father of microbiology. There was a time that people believed in the theory of spontaneous generation, which said that some form of life can suddenly appear out of decaying material like rotten meat. At the time, people were struggling because wine turned sour. In 1856, Louis Pasteur discovered that in the living organism called a yeast that turned beet juice into alcohol. Moreover, he discovered that in the spoiled alcohol, there is another microbe which differentiates called Microderma aceti. This microbe, which is commonly used to produce vinegar, make the wine spoil. This discovery led to the Pasteur's germ theory of fermentation. By experiment, Pasteur discovered that heating the fermented wine could kill the pathogen that make the wine spoil. This process has been used for a long time for beer and vinegar. However, Pasteur patented the process because he pointed out the exact time and temperature that would kill the pathogen without worsening the quality of wine. Before we're gonna talk about pasteurization, we have to know that it is one of the thermal processing. The term thermal processing applies to a range of heat treatment used for food processing. Compared with ultra-high temperature, briefly UHT and sterilization methods which killed all microorganisms in the food. Pasteurization is the milder form of thermal processing which kills only some of them in order to maintain the texture and the characteristic of the food. Even we say that pasteurization is a kind of thermal processing to kill or reduce the number of pathogens. But what is the mechanism behind it? The point is that when the temperature gets hot enough, the enzymes in the bacterium are denatured, meaning they change the shape. This change renders them useless. They are no longer able to do their work. The cells simply cease to function. Heat can also damage bacteria and cell envelope. Proteins and fatty acids making up the envelope lost their shape, weakening it. At the same time, fluid inside the cell expands as the temperature rises, increasing the internal pressure. The expanding fluid pushes against the weakened wall and causes it burst, spilling out the guts of bacteria. After the death of the pathogen, we're gonna talk about two types of pasteurization. The first one and the other one is called wet pasteurization. In the case of milk, for example, they heat milk up to 63 degrees Celsius in the big container for 30 minutes and constantly stir it. On the other hand, the new and common method called high temperature short time, briefly HTST pasteurization. The process is a bit complicated, but in summary, milk is heated and cooled in the range of 4 to 72 degrees Celsius several times in a short period of time. Just imagine what would happen if this world had no any pasteurization. How would you do with your milk and your beer and your wine that you bought? You have no idea how many pathogens and what kind of pathogens are there. Thanks to Louis Pasteur and his innovation to make our life better and easier today.